Thank you everyone for joining me for this brief presentation. My name is Peter Fung and I'm the Senior Manager of Customer Marketing at Nanotemper Technologies. I'd like to talk to you about how you can improve therapeutic monoclonal antibody selection by knowing protein stability first using Prometheus. So as many of you know, monoclonal antibodies are an important class of biologics. Since 1985, the FDA has approved more than 100 monoclonal antibodies as commercial drugs. They've been used for the treatment of cancer, a variety of different inflammatory and immune diseases, as well as many neurodegenerative disorders. Monoclonal antibodies have also given rise to many other different biologics, such as antibody drug conjugates, fusion proteins, bispecifics, a series of different vaccines, and more recently, cell and gene therapy techniques. However, developing and bringing a biotherapeutic to market can be a very long-term commitment that can be from basic research all the way to FDA approval and launch of a product can take anywhere from 10 years to longer in order to achieve this step. Now, in a recent publication by Daunting et al. In Nature Reviews Drug Discovery, they mentioned how essentially 50% of all compounds that make it to phase 3 trial actually end up failing, and that there's about a 10% success rate for compounds entering phase 1 trials. And over these last 2 decades, these success rates really haven't improved. Therefore, developing and bringing a biotherapeutic to market can really be a challenging task. So I would actually challenge you all to think about that maybe stability is the critical quality attribute that should be monitored when developing a biotherapeutic. If you think about this, a protein that lacks stability really leads to the death of that protein. When a protein's unstable, it starts to unfold. It loses its higher order structure. It'll then start to aggregate, fall out of the solution. It basically loses its function or any of its activity, and ultimately it's degraded and broken down and cleared from the cell. However, if a cell is stable, it's likely going to be functional, and therefore you're able to perform research on it, you're able to characterize it. If your protein's stable, it's developable. You can further optimize or improve the protein. And if your protein is stable, it's going to be commercial. You're able to publish on it, and you're able to develop new and novel drugs. Therefore, monitoring protein stability throughout all your workflow steps will really lead to more successful characterization of your biotherapeutic targets. This includes early steps such as discovery and characterization of your protein when you're trying to confirm its structural and functional activity, to various such as screening a variety of different buffers or comparing different batches of material to check if you're able to produce this and purify it, as well as to those researchers that are in production where they're monitoring different storage conditions and really trying to optimize the formulation conditions, and those scientists that are working in manufacturing where you're performing very detailed stability tests, long-term stability or accelerated force degradation studies, as well as those researchers doing the QC on the fill and finish product. So with that, I'd like to introduce to you Prometheus, which is the new gold standard for precisely characterizing protein stability. With Prometheus, it can make precise measurements with high inter and intra-assay repeatability, therefore giving you the highest quality data in your results. It's a label-free detection method in that we measure the intrinsic fluorescence of any protein. It utilizes only a tiny amount of sample, 10 microliters, in order to generate some data. And it has very flexible throughput and is amenable to automation. With Prometheus, our researchers are looking at a wide array of different targets, such as monoclonal antibodies, antibody drug conjugates, biosimilars, functionalized antibodies, and nanoparticles. We have many researchers that look at viruses, virus particles, and virus-like particles. And we have also many researchers that are working with either recombinant or the native proteins and a variety of different types of enzymes, such as kinases. We also have many researchers that use Prometheus to look at membrane proteins and GPCRs, which are often very difficult to study their stability in with other technologies. There's an ever-growing community of over 400 leading biotech, pharma, and academic labs that have made Prometheus their gold standard method for characterizing protein stability. But don't just take my word for it. I'd like to share with you a couple quotes from some of our users. Dr. Thomas Schubert from Tubind, the CRO in Europe, shared with us, the unmatched resolution due to the high density of data points is very beneficial for antibody engineering projects. Multiple transitions can be identified and determined with high accuracy and reproducibility using Prometheus. And Donnie McKenzie from BioMarin Pharmaceutical here in the Bay Area shared with us, we continue to challenge Prometheus with complicated proteins and formulations. It keeps giving us reproducible, high quality data that allows us to get high quality therapeutics to patients faster. Isn't that why we're really doing research, is to develop the highest quality therapeutics and drugs and being able to get those to patients that need them faster and more efficiently? So let's talk a little more about Prometheus. Prometheus runs nano-DSF technology, which nano-DSF technology measures changes in intrinsic fluorescence when a protein unfolds during a thermal or chemical treatment. So a protein will have some sort of folded state, which we can measure. And as you apply either a temperature gradient or a chemical gradient to that sample, will cause the protein to unfold. With Prometheus, you can get very precise measurements in that samples are loaded into capillaries, a small volume, 10 microliters to be exact, is taken into the capillary. With Prometheus, you can test up to 48 samples in a single run, and the user can define the thermal ramp that's applied to the sample. This can range from 0.1 degrees up to 7 degrees C per minute to generate an unfolding curve. And of course, you can run a 1 degree C per minute thermal unfolding to get a true TM measurement. As I mentioned, Prometheus measures the intrinsic fluorescence of your protein specifically at 330 and 350 nanometers. With Prometheus, you can also easily detect the onset of aggregation using our back reflection technology. There are multiple options that address your research needs with Prometheus. The NT48 provides you the most flexible throughput options, allowing you to test multiple conditions in a single experimental run. With the Prometheus NT-Plex, it's more for scaled up and organized higher throughput of your sample, in that it utilizes chips that hold the capillaries. In these chips, there's 24 in each chip that can be run in the experiments. And then in addition, you can add the NT robotic autosampler to get a fully automated hands-off solution. So let's talk about some data examples. As I mentioned with Prometheus, you can get deeper characterization of your monoclonal antibodies using simultaneous detection of both conformational and colloidal stability. What's shown in this example here is a monoclonal antibody that's subjected to 6 different pH conditions, pH 4 all the way up to pH 6. As you can see, the different conditions impact the thermal unfolding of the sample, where pH 4 makes it the most unstable. If you looked at this data on its own, you would probably say, well, actually, I should probably be working with this pH 6.5, as that seems to keep my antibody in the most stable condition. However, with Prometheus, as I mentioned, you can also measure the onset of aggregation and do this simultaneously as you're doing the thermal unfolding to take both of this information and make a better decision on characterizing your different antibody candidates. As you can see from these results, pH 6.5 actually causes the antibody to aggregate a lot more than the lower pH conditions. Therefore, using both these pieces of information of the thermal unfolding and the aggregation data allows you to make a better selection of which condition is able to keep my protein in a more stable state. With Prometheus, you can perform elaborate screens testing multiple variables within the same run. And this example here is just showing a wide array of different detergents that are used to unfold this antibody target here. As you can see, a wide array of different conditions and unfolding profiles are generated with the different buffers, the detergents that is. So therefore, you can screen a wide variety of different detergents, select the ones that keep your protein in its most stable condition, and then focus your efforts on further optimizing those conditions. With Prometheus, you can also screen for secreted antibodies in cell culture supernatant. What's shown here, as many of you know, as you're generating monoclonal antibodies, you'll generate the hybridomas that secrete the antibodies into the supernatant. What's shown on this right panel here is a IgG that's spiked into PBS to show you the unfolding profile that's generated. There are two transitions that are detected. And then the supernatant material collected of the same antibody producing hybridoma. And you can see those same unfolding transitions. With Prometheus, you can even take this one step further. If you know the antigen that binds to your antibody, you can perform a label-free thermal shift experiment. What's shown here on the left panel is the antibody by itself in the supernatant. You can see there are two unfolding transitions. When you add the antigen to that, you actually get one unfolding transition that's detected. Label-free detection, very quick check as you're screening your different hybridoma candidates. Using Prometheus allows you to quickly identify the top lead candidates in your analysis. Last example, I just wanted to show you how with Prometheus you can detect very discrete and unfolding transitions that help you distinguish your different antibody candidates. What's shown here are three different antibodies. You can see the unfolding profile in the ratio view on the left, as well as the first derivative view shown here on the right. As you can see, there's a nice unfolding transition. And when you look at the first derivative view, you can see the first transition, the second, and the third transition here. And you can distinguish between antibody B and antibody C, the unfolding profiles of the first and second domains as they're unfolding under the thermal treatment. In this particular example, we just want to mention again with Prometheus, a delta Tm of 0.2 degrees is considered significant. With Prometheus, we can really help you address the different stages to optimize a biotherapeutic production workflow. Early steps involved with the expression of your protein. You want to test and see, can I identify my clones? Are they being expressed? When you're working on the purification, it's important and critical to monitor the stability. You can test the thermal stability as well as the aggregation of your protein in the various formulations. You can even, as I showed in the example there, do a quick functional test doing a label-free thermal shift analysis. If you need to do deep characterization, Prometheus is perfect for this, to look at optimizing fragmentation conditions, as well as testing the effects of post-translational modifications on the stability of your protein, as well as the onset of aggregation and potentially its functional activity. And then finally, in the later stages of your workflow where you're doing assay development, you can use Prometheus to optimize those experimental test conditions and really do a deep dive on looking at the functional activity of your protein. So Prometheus really is the new gold standard that precisely characterizes protein stability. With Prometheus, you can precisely measure the conformational and colloidal stability of any protein. It's a label-free detection method, and it uses only 10 microliters of sample to generate your data. You can measure over a wide concentration range from 5 microgram per mil all the way up to 250 mg per mil within the same experiment. And our technology, we like to think of it as being buffer and detergent independent. For those of you who work with a lot of membrane proteins and GPCRs, you know that you need to utilize detergent in order to study those types of proteins. Those aren't a problem at all with Prometheus. However, with other technologies, those may actually kill the readout of your sample when you have to do this in the presence of high detergents. And as I mentioned, with Prometheus, you have flexible throughput options and the ability to automate your samples. So really just want to conclude by saying that at Nanotemper, we're focused on helping you, the researchers, find solutions to help better characterize your proteins. Today I was talking about Prometheus, which is used for precise characterization of protein stability, but we have a wide variety of other tools to look at characterizing your proteins. We have Tycho, which is the everyday solution for a quick check of protein quality. Monolith, our flagship system, is really the choice for measuring molecule interactions. And we just recently launched Dianthus, which is the high throughput solution for measuring binding affinities. So if you have any questions, please feel free to reach out to me at my email address shown here, or visit our website at nanotempertech.com. Love to be able to hear from any of you if you have any questions, and please check out the website and learn a little more about what we have to offer and the various tools. So thanks again for taking the time, and I hope to hear from you soon.